Okay, so are you ready?
Yeah.
Okay, so good morning, afternoon, evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you for joining us the 40th seminar. Oh my God, already 4 years seminar, and I call it NIH Day. I am joining SynBYSS from Cancun, Mexico, after my 17th talk in an ECI conference. I discussed plastic upcycling and climate crisis, as well as probiotic engineering and biocontainment using kill switches during the conference. I have 4 more weeks out of the 10-week world seminar tour, but my stay at Cancun has been super nice, as you can imagine. Okay, so it is my tremendous honor to introduce very briefly our pioneer speaker today, Dr. David Rampulla. He is the program director at NIH, who has been supporting many synthetic biology researchers, including today's rising star speaker, Professor Gao. So we thank him for his commitment to the community and support. And what else do you want to know other than his important role that he plays? So Dave, thanks so much again for your guidance and support for synthetic biology community members, and the virtual podium is all yours. And thank you so much.
Right. Give me a couple seconds here and I'll share my screen. Just got a couple of slides. Okay. Well, so first, thank you. I don't know that I'm a pioneer, but I appreciate the honor. Yeah, I've done what I can to bring synthetic biology to a couple corners of the NIH, but it wouldn't have worked all without all of you. So thank you for keep coming. I know not everyone gets an award the first time they apply, so I really appreciate your persistence. And no doubt over the last 5-6 years, synthetic biology has grown around the NIH. And so I just thought I'd take a couple minutes here and just kind of hopefully give you a couple little helpful tidbits. And so you might think about bringing your work to the NIH too. So first thing I just wanted to mention is that there's a notice of special interest out there on the street right now, synthetic biology for biomedical applications. It's really broad. And I'm really excited about this because to kind of demonstrate how things have grown at the NIH, we had a similar announcement out in the street a few years ago that had 3 institutes and centers on it. And now it's grown to 8. And so, you can see there's the listing, NIBIB, right? We're biomedical imaging and bioengineering, cancer, NCCIH, complementary and integrative health, general medicine, NIAID, aging, genomics. So it's, it's a pretty broad cross section of about 1/3 of the NIH that has demonstrated a specific interest in synthetic biology. So yeah, if you think some of your work might fit into one of these buckets, you should totally look at this notice. And within this notice, there's lots of different funding opportunities there. So there's the parent R01, the NIGMS MIRA is part of this. So I know that's a pretty exciting mechanism right now. And I think they've supported a few synthetic biologists through it. There's a couple R21 mechanisms in there as well, including the Trailblazer at NIBIB. So I think there's a lot of good opportunities for the young synthetic biology set that might be tuning in today. So so check this one out, please. I guess I thought I'd just, so any of you that know me out there, I probably work this into every talk I ever give. And if you can walk away from this, the one thing I would hope you remember is contact a program director. You should do this before you apply. We're super nice and pretty competent, and we can help you. So because there are differences between the missions around the NIH, and if you apply to the wrong institute and it's a mechanism that we can't transfer to somewhere else, it just goes back to you without review. So yeah, please, like, we're eager to hear from you, and we're here to help. So yeah, reach out to a program director when you're thinking about applying. And I thought I would just make a quick plug for NIBIB. We like to think of ourselves as the engineering IC, right? We really focus a lot on developing modular platform technologies. We're disease agnostic. And I think there's just a really natural fit between NIBIB and a good cross-section of synthetic biology. So yeah, so please, think about NIBIB if you're interested in engineering a new modular SynBYSS technology. And my last slide, just to kind of highlight a couple key opportunities out there for the trainees. So the first one here is the KO1. It's a mentored research award. This is an interesting one. It's a supervised career development award. And in particular at NIBIB, we use it for people who are looking to get into a new field. So while this one may not be something that current synthetic biologists might want to use, I mean, they could use it to go into a non-synbio field. You could also tell your friends about this one if they're actually interested in coming into the synthetic biology field and mentoring with you. So this could be an interesting way to make more synthetic biologists. So the KO1, pretty cool award mechanism. I think a lot of you probably are aware of the F32 if you're from the NIH space. It's just a really good, solid postdoctoral fellowship. So don't forget about the F32. And then the last one I'll mention, which is super important to me today, because your next speaker is a K99 R00 awardee in my portfolio. And so it's really awesome, right? You get, it's a mentored postdoctoral position that transitions into your independent career. And I won't, I'll let Dr. Gao talk about it if he wishes, but I think they're pretty helpful. And so if you've got some pretty outstanding ideas, the K99 R00 is a really great mechanism. So yeah, I think that was about 5 minutes of me yammering on about something. So I hope you got a couple key messages out of that. And I've got my information down in the corner. So please reach out to me. I'm happy to chat and we'll take it from there. Thanks so much.
Thank you so much. That's fantastic talk and very useful information. I mean, especially for young people, they sometimes very shy and then not that much willing to reach out to program manager, but they now have some courage to contact you. So thank you so much. And I completely agree with you. I hope you will continue to support the community, especially young researcher like Dr. Gao, got previous years. So thank you so much for your time and commitment.
Thank you.
Yeah. Okay. So now the main speaker of today with a longer introduction, Professor Xiaojing Gao is an assistant professor of chemical engineering from Stanford University. He received a PhD in biology from also Stanford University, building genetic tools to study neuroscience. He was then converted to a synthetic biologist for his postdoctoral training, working with Professor Michael Eloich at Caltech, of course, one of the synthetic biology fathers. His lab addresses fundamental engineering challenges across different levels of complexity, including protein components that minimize their crosstalk with human cell and immunogenicity. Second one is the biomolecular circuit that basically function robustly in different cells and are easy to deliver. The last one is basically control circuit that counter the delivery stochasticity of conventional gene therapy. The engineering targets include biomolecules, molecular circuit, viruses, and cells, and his research combined quantitative experimental analysis with computational simulation, and actually not many people kind of doing both together in the same lab. The molecular tools he will be applied to immunology, neurobiology, and cancer therapy. He's the true rising star with the very, very bright future, and I'm so glad to have him today as our rising star speaker. And Dr. Gao, please take it away, and thank you so much for joining us today and a little bit early time in your California time today.
Yeah, no, thank you, Tae-Suk. And actually, I'd like to say a few words before I share the slides, right? First of all, thank you. I mean, many of us complain that there is no platform for younger people, and then we are just complaining. But it takes your dedication and vision, right? Basically, you single-handedly started this. And that's awesome, right? For young people like me to talk, but also to have good excuse to not change diaper for 1 morning. I'm just contrary. So that's good. And then speaking of single-handedly and vision dedication, I think these labels also apply to Dave, right? In the sense that for even younger people than me, they probably don't even remember back then when SynBYSS was mostly supported by DOD or DOE. And a few years ago, there was this young program officer who had this vision. He's like, maybe SynBYSS can make a difference to human health, despite us telling NIH for many years. And that's what's the difference. As you can see on the nosy, now all these different ICs with NIH are signing up. And that's great. And from a more personal perspective, I think I had a misfortune because Dave was the very first PO I ever contacted. And that set the expectation not quite right? So don't take me wrong. Every PO I worked with are great, but there's also exceptional POs like Dave who are responsive and engaging, right? And now you are promoted to a director, but you still keep that style. I don't know how you manage that, but I'm not complaining. So yeah, I think I want to echo Tae-Suk's comment that it's really fortunate for synthetic biologists to have ally like you within NIH. And I'll just leave it there. And
so that's why I call him pioneer.
That's right. That's right.
Period.
I literally tell my
Thank
students,
you for the kind words.
If you want to make an impact, you can either do great SynBYSS work and run a lab, or you can go be like Dave. I sent some of my collaborators to interview for Dave's the PO position precisely for that reason, because that does matter, right? And also I want to thank the 3 people who show up. I'm very used to this because teaching during during the pandemics, right? Like undergrads sometimes don't show up, so I actually feel very at home talking to 3 people and knowing that many others will be learning asynchronously later. But to reward the 3 people, please feel free to raise your hands or any way to interrupt. If I didn't see your hand raising, maybe Tae-Suk can help remind me there is some question in the middle of the talk. I wouldn't mind at all. Yeah. All right, now screen sharing. Can you guys see? Yeah. So I'm not going to introduce myself, but speaking of teaching, right? You may notice in my background, there's no chemical engineering education. I was more like a biologist, but I have to teach chemical engineering, such as reactor design. So this kid asked me, they're like, Xiaojing, what if my reactor is contaminated? And I was like, go be a biologist. You take some bleach, you spray this all over, and some of those will miraculously seek into the reactor and fix it for you. And you don't have to be a chemical engineer to see how ridiculous that sounds. But if you think about it, that's what happens every day. When one of us gets sick, you take a medicine and hope that some of the medicine will leak into this reactor, your cell, and fix things for you. Okay? So not to be disrespectful to the traditional pharmacologists, despite this fundamental challenge, they saved many lives already, which is amazing, right? But our assertion is for certain diseases, it's just too complicated for this approach to work. Instead, we want to use circuits as medicine, right? So these circuits are metaphors for biomolecules engineered to regulate each other. You can introduce this entire set into the cells so that they can detect the state of the cell process the information and then actuate therapeutic outcomes. And one example could be, if the circuit knows this is a cancer cell, and circuit can then actuate suicidal outcome, or even better, it can engage the immune system to destroy other cancer cells. And you may wonder, if this is as rosy as you claims, and you can hear my family screaming in the background, but if it's as amazing as it seems, why haven't we seen much or any synthetic circuits as medicine? And the reason is, we're still in the foundational building stage. The building blocks are not all there yet. And that's what my lab is working on. On one end of this, we're trying to, let me turn on the laser pointer. Because historically, most of the components people use in synthetic biology come from non-human organisms. And the fundamental problem is the moment you use that in a biomedical context, human immune system will recognize them and try to interfere with them, or worse, have cause adverse effects. So we're asking ourselves, can we recreate some of the most important tools in synthetic biology, but also make them masquerade as our own components so that the body does not overreact? Right? So that's one wing of the lab. And the other wing is we're also trying to, although there's a whole field working on gene delivery and gene therapy, we believe we have our unique perspective, so we're trying to do something different here. But today I'm going to share mostly with you two vignettes in the middle part for which we have made the most progress. And most of them actually goes to this we're at an exciting eve of, or maybe already we're in the age of RNA therapy, right? Many of you know this, and I suppose most of us have this at some point injected into our cells. So the one fundamental challenge, I'm sorry I have to mute because there is some mishap in the childcare. I have to be back in 1 second. Yeah, if there's a Chinese speaker in the future audience, they will know what we're talking about. So where was I? Yes, RNA therapy. Right. So for synthetic biology, one fundamental challenge is for these circuits to work, you have to deliver them into human cells. And that sounds risky and difficult, except that in the context of RNA vaccines, it has been demonstrated that you can do such mass production, distribution, and administration on a global level with very low safety concerns, right? So, basically this could be a future vehicle. And in addition to RNA vaccines, people are also developing RNA for other kinds of gene replacement or other therapies. Except that the current tools in synthetic biology are not quite compatible with this, and Tae-Soo's canonical work on all these logic gates in multiple cells, right? These are amazing for engineering bacterial populations. But what they have in common, I would say 95, maybe 99% of synthetic biology work is to rely on the use of transcription factors. And transcription factor have to operate on a piece of DNA, but if you deliver with a piece of RNA, they just won't work, right? So this is the challenge where I'm trying to address with the two starters I'm sharing today, but this started during my postdoc. So back then, we had this idea that if all the components of synthetic biology are proteins, and if they talk to each other directly at the protein level, it wouldn't matter, right? Whether you deliver with RNA or DNA, they will still operate most likely robustly. But back then the challenge was how can you make these protein circuits without too much engineering effort? Because unlike transcription factors, each protein is quirky in its own way. It's not really scalable, building a sizable protein circuits. So our little epiphany was we can take these viral proteases, and they are orthogonal enough, but they're also similar enough. So if you engineer one of them, you can similarly engineer all of them. And with a small set of them, we managed to demonstrate a broad variety of applications, right? So that's the work. But there are still a few key missing links. One of them was to do biomedical applications, such as engaging the immune system. You also have to regulate intercellular communication. However, that work we published only have intracellular circuits regulating intracellular proteins, right? So the question here is, can we use an intracellular protein circuit to regulate the communication between cells? And that's when Dr. Alex Vlahos joined my lab. And by the way, I'm super proud because Alex is going to be a SynBYSS presenter in a future talk, so stay tuned. And in that spirit, I'm going to make this part of the story shorter to not steal his thunder, right? But Alex has his background in tissue engineering, and he is very interested in how do we use SynBio to modulate the host immune system to make it more friendly to the transplanted tissues. And then, to do this, he looked at the style of work in my lab is we usually start by asking how does nature do, right? So there turns out to be a very tightly regulated transport pathway going from endoplasmic reticulum to Golgi apparatus and to the cell surface. And some of the proteins have to really remain in the ER or in the Golgi, and there are specific signals for doing that. For example, these proteins, that has a COP1 recognition signal as identified here, they'll go to the Golgi, but then retreat back into the ER so they can never escape. Oh, and by the way, I should also mention, Jenny Kang has made a major contribution to this project as well, and with some support from Carlos already, both students in my lab. So Alex, look at this, and here's the design we came up with. I already see two chats. Hope I didn't miss any. Yeah. So design look like this, and I'm gonna try to walk it slowly, but please ask me to repeat if it doesn't make sense. This is a signal protein you want to regulate that will eventually go into the extracellular space, but not yet, because it's covalently fused to a bunch of transmembrane domains, and then eventually retention signal that tells the protein to stay within the ER. But you can either use multiple transmembrane domains or one of them, but the key is somewhere in that transmembrane domain and the linker region, you want to have a cleavage site that's inserted so that when you have a protease, you can cleave here or here, right? And now the secretion part is physically separated from the retention part. This part is still retained, but this part can now go on from ER to Golgi and onward. And the other key part is we also have a second cleavage site for furin protease. Furin protease is endogenous protease. It's not present in ER, but it is present in trans Golgi. So that once this whole construct is in trans Golgi, there can be secondary cleavage, and the secreted protein can be separated from the membrane tethering to finally get into the extracellular space. Does that make sense? Yeah. Right. So now does this work? And also want to mention, around the same time there is we published this earlier this year, but around the same time there's two other papers from Martin Fusenegger and Roman Gerard's lab, which used a kind of similar philosophy, protease dependent ER retention to regulate protein secretion. So check them out. Okay. And to characterize and optimize the system, we use an enzyme called SynBYSS. It's relatively easy to measure its activity in the supernatant of cell culture. And as you can see here, we make this construct regulating secretion in the absence of the protease, very low signal, and in the presence, the signal goes way up. We can also do a small tweak on the design, which is removing the cleavage site. And now, if you release this from ER, it still goes to the cell membrane, but stays there and does not go further into a free soluble mode. And we validate that it still works using GFP as a model self-surface marker, right? With protease, the surface display of GFP increases. And it's also a modular design. It's exactly the same design. We just took a different protease and used a different cleavage site for the protease, and now signal goes up according to this different orthogonal protease. And in SynBio, it's in SynBio you cannot get away with not doing logic gates, but it's also not frivolous because sometimes you do have to respond to multiple inputs to have more accurate response, right? Or logic is fairly straightforward. You just have to have two cleavage sites for two different proteases, and the presence of either protease alone would suffice to secrete this protein. As you can see here, no input, single, double inputs. And end logic is probably even more useful, right? Because sometimes a single input is not precise enough to define the response you want. And you want the response to depend on simultaneous presence of two inputs. So here what we do is we find out a separate retention motif that you can put on end terminus. And it goes like retention signal, cleavage, transmembrane domain, two furin sites flanking a cargo, another transmembrane domain, another chi site for orthogonal protease, another retention signal. And as you can see, cutting either would not be enough because there's the other retention signal. You have to cut both for a signal to get secreted, and that is validated, right? Single inputs, no increase of signal. It takes double inputs to increase the signal. And for the interest of time, I'm not going to go into how exactly we do this, but we just suffice to say we have finally built a circuit that I alluded to multiple times in the introduction, which is the circuit that responds to the presence of oncogene. It's actually a notorious oncogene called KRAS, the undruggable KRAS. And that's through a relay protease, because this happens at the cell membrane, and our release, our controlled secretion happens at the ER membrane. So we have to have a relay protease that sends the signal from the cell membrane to the ER membrane, and eventually use our release mechanism to regulate a cytokine that actually people inject this cytokine locally into cancers as a potential therapy, right? So we built this whole circuit and we validated that it does respond to the presence of oncogene. And again, I apologize for not going into details, but after all, it's all published work. So if there's any interest, we can always go into that, right? And what is more, this sense, relay, and response topology is actually quite modular and quite versatile, I would like to brag, because there's a whole field building all kinds of receptors, converting extracellular and intracellular inputs into protease activities. In our previous work, we have this chunk platform that can process information, and the release itself have some information processing capability. And after that processing, we will actually demonstrate we can regulate all kinds of inputs. It could be secreted cytokines or model proteins. It could also be cell membrane proteins such as ion channels, right? And we sort of picked a few of these input-output combos, and without exception, although quantitatively they may differ, but qualitatively every construct every circuit we constructed using this release platform seems to have worked, and that made us quite excited about. Right? And this ends my very brief part about the release platform that regulates protein secretion. If there's any questions, you can interrupt or type in the chat or raise your hand. Yeah. You guys are precisely like my students who are, I don't know. So now moving on to the next part, right? Because one keyword in my previous slide is qualitative. Okay? Protein circuits are great, but at the end of the day, for every different signal to process, it still takes some engineering to adapt to the quirk of that specific protein signal, right? And we'd like to claim the good engineers are lazy engineers who are like, what if there's a scenario that you don't even have to do that? And in that sense, sensing RNA will be easier because RNA comes with this free base pairing, right? So in principle, you design something and you just change the base pair, then you can sense something else. But can we do that? And this work has been led by Eric Kazenit, a grad student, and also with major contributions from Dr. Noah Katz, Dante Kober, and Connor Cole. Noah is a postdoc, and Natalie and Connor are students in my lab. Right? And there are other reasons to detect RNA as well. First of all, there's explosion of RNA sequencing data, or specifically single RNA sequencing, to the point that now for any cell type of interest, if the data is not already there, it's almost trivial to obtain its RNA profile. Right? And then if you ask the right people, like Steve Quake, they will tell you, once you know the RNA profile of the lab, you know a lot about what the cell is doing. And maybe precisely because of that, not surprisingly, around the same time we put our preprint out for the next story, there are at least 3 preprints out there using a very similar design philosophy to create RNA sensors. Right? So the first one is from Boston. They had a lot of exhaustive and impressive engineering work. The second one is from Peking University. And the third one is from Duke, Josh Huang's lab. And they took it all the way actually to human cortex, demonstrating this design seems to be compatible with all kinds of biomedically relevant situations. And you may wonder, why all this excitement? What can you do with these RNA sensors, right? So basically what you want to achieve is, in the presence of any specific RNA input, you want to convert that to arbitrary protein output all within living mammalian cells. And you can use this for basic research. You can look at how transcripts change over time, such as during development, when genes get upregulated, downregulated, or during an infection, when there are different subgenomic species of RNA virus that it's impossible to trace with traditional tagging approaches, but maybe you can use a sensor to convert that to outcome that's easy to monitor. Or you can use this not just for observation, but for manipulation. Again, in a basic biology context, if you can use this kind of sensors to specifically change the activity or ablate subtypes of neurons or immune cells within a complex network, that's probably the only way you can establish causal relationship between these type of cells and their function in the larger context, right? Or in the more medical context, if you can specifically detect markers that represent cells you don't want, cancer cells or aging cells, and you specifically ablate them, and that's potentially a precision therapy beyond the whatever has been achieved with traditional methods, right? But the question always is, right? Repeating the mantra I repeat many times today, this all looks great. Why haven't we seen good RNA sensors? And when we talk about RNA sensors, your mind usually immediately goes to toehold and the strand displacement strategy it represents, right? It's been almost a decade now. Again, without going into the details, this is from Yingpeng's lab, but the basic idea is for a strand displacement sensor to work, you design a piece of RNA sensor. It's designed in such a clever way that once it forms double strand with the input sensor you want to detect, there's some conformational change in the design sensor, and that conformational change leads to the up or down regulation of some protein translation or expression, right? But it took people quite some time until very recently, this paper from Jim Collins' lab, and there are also related work from Georg Selig's lab and Niles Pierce's lab, right? It really took people a long time to take these strand displacement sensors that seem to work amazingly in E. coli to mammalian cells. And I happen to know some of these authors, right? And so I know much more about the underwater part of the iceberg that didn't even make it, all the sensors that's very hard to tune in mammalian cells. So when we enter this field from a different angle, looking for RNA sensor, we ask ourselves, why? Why is it so hard to build RNA sensors in mammalian cells, and can we do it differently? And we reason that's because in mammalian cells, we have evolved a whole suite of double-strand RNA engaging proteins, because double-strand RNA does not exist naturally in human cells, and their presence signifies viral infection, right? So we have all these double-strand RNA engaging proteins, and strand displacement strategy rely on the formation of double-strand RNA, but that did not take into account these endogenous human proteins, right? So these sensors will be interfered in unpredictable ways, and maybe it's not too shocking that it's very hard to make them work. And now we look at this situation and we decided that there's really no way to go around these double-strand RNA sensing proteins or engaging proteins. So we're like, can we turn this bug into a feature and use them as a sensing mechanism? And based on that philosophy, we quickly zoomed in on this specific protein called ADAR. So ADAR is a housekeeping protein that's present in all our cells. Their natural function is they bind a double-strand RNA, right? And then they added the adenosines there to inosine. Okay, A to I editing, and I in translation is treated as G. Okay, so that's a very specific thing they do, which is always good if you want to engineer something specific is good. And there are additional useful features. First of all, there does not seem to be a strong sequence identity preference, meaning ADAR just likes to bind double-strand RNA, but not certain kind of double-strand RNA. Right? So that makes the programmability easier. And also, editing by ADAR seems to bias the cell response away from the other double-strand responsive pathways. So it's also sort of naturally isolating your engineered sensor from interfering with and being interfered by all these other pathways. And there's also this engineering benefit, which is in a different context, in ADAR-dependent editing of endogenous transcripts as a potential gene therapy. There are a whole field, especially from the Stanford, Wei, and Mali labs. And so they have figured out a lot of engineering tricks that we can borrow for our sensor. So the question, how do we convert this editing event to a sensing mechanism? And this is what we came up with, right? So now this is the endogenous RNA, the marker RNA you want to detect. And from now on, I'm going to call this the trigger RNA. Right? So to design a sensor RNA, it's a single piece of RNA with 3 segments. It starts with an optional co-transfection marker, followed by a key stretch that's designed to be reverse complementary to the trigger RNA. And the feature is in the middle, the in-frame stop codon UAG. And it's designed in such a way that when a double strand is formed, it will be positioned opposite the CCA so that the editing of the A by ADAR is favored, right? And then there's the output. So in the presence of this sensor RNA alone, the translation starts and stops here, no output. But once you have the trigger RNA, you get double strand, A to I editing, stop codon becomes tryptophan codon, translation can go through, and GFP is turned on. Okay? And this doesn't have to be GFP, it could be anything. And we also use two-way peptides, self-cleaving two-way peptides, because this part is variable, right? So we want to insulate this variability away from these other markers, right? I'm checking chats again to see if there's any questions. Does that make sense? And we made this key design decision, which is for the sensing region, we want to prioritize 3' UTR because we reason that for the double-strand RNA region to be stable, we want to avoid actively translating ribosomes, which again could happen, right? The ribosome could interfere with the double-strand RNA, the double-strand RNA could interfere with ribosome. And furthermore, in addition to the intended stop codon editing, other adenosines in double-strand region also have the chance of getting edited. So we want this editing to happen in the 3' UTR that's less likely to introduce a detrimental copy of the host protein versus if you edit them within the coding sequence of the host transcript. Right? And this design seemed to work as we put these components. And by the way, all these characterizations, although I drummed up the RNA aspect, all these initial characterizations were still done using plasmid DNA encoding these RNAs just to make the design build test cycle easier. So what we did here is, here's the sensor. This is a mismatch trigger that's not supposed to trigger it. And this is a match trigger that's supposed to, and that's all consistent with expectation. And also to make sure that the sensors are operating as intended, we overexpress ADAR, right? In addition to the endogenous ADAR, and indeed that overexpression seems to boost up this trigger-dependent signal. Right? So this both validates the intended mechanism, and we have other work that I'm not sharing, like knocking out ADAR, et cetera, all as validation. But also ADAR overexpression becomes a way for us to consistently boost up the signal of the current version of the sensor. And finally, to estimate how good the current version of the sensor is, we made a positive control. So the positive control is basically a sensor sequence, except that this A is already made into G. So every copy of RNA should have the expression of downstream EGFP. And that's about 33 times higher than the high signal, which means about 3% of the sensor is edited in this most favorable condition, right? A number that will be useful when you actually think about using these sensors, and also tells us there's plenty of room to improve for future generations of the radar sensor. And then skipping over a bunch of characterizations and optimizations, the question that everybody asks is, up to this point, we're using synthetic triggers, right? We just transfect another plasmid that makes a ton of the trigger, but can you detect something that's endogenous? So we first tested GAPDH. This is like your internal qPCR control. And this is not sensor performance yet. This is just using qPCR to see whether we can get different levels of GAPDH using different amounts of RNA knockdown, right? And then in these exact conditions, we introduce our sensor and we measure the GFP output. And we see, indeed, there is the same trend, right? Increasing amount of output as you increase the GAPDH level, although the relation seems sublinear, right? The fold change is always smaller in the sensor case than in the qPCR case. And another example we tested for endogenous type detecting is a heat shock response. And so there again, we build a sensor for a heat shock gene. And this is the first thing you want to look at, right? So this is a sensor response. This is a sensor output in response to heat shock. But just to make sure that there's no other non-specific fact, we have two types of controls. One is a mismatch sensor. So this sensor is not supposed to respond to that heat shock transcript. So we see there is a smaller heat shock non-specific response, but smaller than this. And the other control we did, we also have sRNA that knocks down the gene that this sensor is supposed to be sensing, and that also reduces the amplitude of the response, right? So after we do a three-way ANOVA on all these factors, there's a significant interaction, which means there is a specific, even after you account for the non-specific heat shock effects, after you compare this condition to the knockdown condition and to the mismatch sensor condition, we are confident there is a specific sensor response to this heat shock gene. And the heat shock gene level is at about 7% of GAPDH transcript abundance, right? So that's why where we estimate, somewhere that is the sensitivity of a sensor right now is 7% of GAPDH. And there's more work to do to make them back. Now switching gear a little bit, one thing we're really curious about is how many different types of transcripts we can detect, right? Because that, after all, that's the premise of the whole project. We want to be able to program the sensor for as many different inputs as possible. And we tweak a few knobs to make the potential candidate pool bigger. So the first thing we did is the shorter the double strand are. So one limitation is, in addition to this stop codon, you do not want to have other in-frame stop codons that's accidentally introduced through reverse complementation. That means the shorter the double-strand RNA region, the less likely that you accidentally get other stop codons, right? The shorter it is, the more candidate targets you can sense. So we alter the length of double-strand RNA and ask, does the sensor still work? And usually we, like in all the data I showed you before, we use 90 base pair double-strand RNA. It can go up to 153 or go down to 72. Doesn't seem to affect the output. Although if you keep cutting it down to 36, now the signal is gone, right? So we know the rough range of functional length of double-strand RNA. And then looking at this, we ask ourselves, can we rescue this loss of sensor output by putting a stretch of double-strand RNA somewhere else, right? So here we have a split design. This is the 36 base pair you just saw in the previous slide. And now we design the next 54 base pair on the sensor that's reverse complementary to a separate region on the trigger. And this is the outcome from this sensor, right? Not as high as a 90 contiguous double-strand RNA, but still obviously elevated. And we really like this design because this will help us skip over accidental stop codons or other features that you may not want, and you can get much higher flexibility in what triggers you can potentially detect. And third thing is, although I discussed early on why we want to favor 3' UTR at the sensing part, we still want to see if can you detect something in the coding sequence at all, right? So in this design, it's exactly the same sensor. We either have the trigger sequence embedded in the coding sequence of the GFP, or we take this same trigger sequence, exactly same trigger sequence, and put this in the 3' UTR of the trigger transcript. And we saw that this is a response to the coding sequence embedded trigger, and this is the 3' UTR trigger, right? So the moral story here is, yes, consistent with our intuition, 3' UTR trigger is better, but if you're really desperate and need to look for trigger regions, the CDS is also an option. And now combining all these improvements, right? Shorter double-strand RNA, the potential to do split, and also triggering from the CDS, we perform bioinformatic analysis on human and mouse transcripts, and they look pretty much the same, right? So for the vast majority of transcripts, there's at least one potential trigger region you can use to design a sensor for. And in 2/3 of them, actually, there are so many, there are like over 50 candidates. And the number is important because right now we still don't fully understand the design rules. And usually we have to screen a few sensors to find one that works the best. And there'll be plenty to screen. Any questions so far? But you may also notice another thing that's all in the figure legends, but I did not highlight, which is we tend to overexpress this ADAR-1 to boost up sensor output, right? And you may wonder, does overexpressing ADAR-1 cause other problems? Because it could also edit the other natural double-strand RNA structures in the cell. And to mitigate that, we took this approach. So a wild-type ADAR has two domains, roughly speaking. It has a deaminase domain that's responsible for the A2R editing, but it also has a double-strand RNA binding domain that's responsible for binding this long stretch of double-strand RNA. So we ask ourselves, what if we take the deaminase domain and fuse it to a more specific double-strand RNA domain from a phage, and at the same time, on our sensor, we can introduce a specific double-strand, actually stem loop motif bound by that protein. And as a result, we expect this chimeric ADAR to only specifically bind to our sensor, but not to other endogenous sensors, hopefully, right? So we build this sensor, and this is a sensor in response to a trigger without the chimeric ADAR, and this is the same sensor with chimeric ADAR, right? So as you can see, also as expected, the baseline goes up a little bit too, but more importantly, the dynamic range is better, and the input and the input dependent signal is much higher than the without chimeric ADAR version, right? And also to confirm that this version indeed reduced its binding to just regular non-specific double-stranded RNA, we took our original sensor, and then we have the trigger, and then we add or not add the chimeric ADAR. As expected, this chimeric ADAR is no longer able to induce the output from this old sensor, suggesting that maybe by using this design, although it does not eliminate the problem, it's likely to greatly reduce non-specific editing of general double-stranded RNA and focus on our sensor. Right? And one thing we noticed in this diagram is we intentionally put the MS2, the stem loop, in the 3' UTR of our construct, right? For the same reason, because we suspect that if you put this in the coding region, the translating ribosome may be cross-interfering with the stem loop. And this might be a good place, to compare some of these design decisions, right? Because what I really like about the Boston paper is they have a very specific design guide. At the end of that paper, they tell people, if you want to build a sensor, do this. And what they do is they say, you put your MS2 loops, very similar to what I just talked about in the previous slide, put the chimeric ADAR recruiting MS2 loops right next to the sensor sequence, right? Which seems to be contradicting what I just said. And actually, curiously, in these other 3 stories, the designers didn't seem to focus too much on the choice of coding sequence versus 3' UTR. But regardless, they did this, and we're wondering, does this matter? Does this design decision matter? And one thing people in the audience might know is it's almost always apples to oranges when you try to compare the dynamic range output strengths from the different papers, because there are always these subtle things people do that makes a direct comparison possible. So instead, what we did is we built a sensor according to their exact design and build the corresponding sensor according to our design. The only difference is the position in MS2. So this is their design. What you can see here is in the presence of a chimeric ADAR, in response to a trigger, this is a signal you get. And in this specific case, actually the signal does not seem to care whether you have a chimeric ADAR or not, as you can see in these comparisons. Versus our design, right? Same design, MS2 now in the 3' UTR. This is the response to the trigger. And this is showing that actually now having the chimeric ADAR does matter. The chimeric ADAR improve the signal. Okay. So this is already too much chest thumping that I feel comfortable in any single talk. But my point is, I'm sure there are certain things that their design are better than ours. And I'm just generally happy that there's this excitement in the field. And I'm hopeful, combining all our design ideas will make these sensors even better in the near future. Okay. And now switching over, from engineering to characterization, I just want to share with you, maybe I have a few minutes left, and a few nice features about this radar sensor. The first is, it's often, as I mentioned before, that one sensor, one marker is not enough for unambiguously identifying one cell population. How do you do the AND logic? And it turns out that AND logic is especially easy given the design of radar, because all you have to do is you can randomly concatenate multiple sensor regions, right, with multiple stop codons. And you can see now it takes the presence of all of these input RNAs to edit away all these stop codons for the translation to go through. As we validated here using a 2-input AND logic, right? It takes both inputs for signal to go on. And the other scenarios that might be useful is, first of all, in a lot of diseases, the condition is not the change expression level of a transcript, but rather change identity of the transcript. Right? For in this example, there are two different dinucleotide mutations, P53. Both are found in cancer, but they seem to be associated with different levels of invasiveness. And because ADAR editing is very sensitive to the base pair base pair matching or non-matching around the editing site, we ask ourselves, can we leverage that sensitivity to make radar sensors that can distinguish this very subtle difference in the coding sequence? And we really could, right? So this is sensor alone. This is with the mismatched P53 mutation. This is with the matched P53 mutation. And so you see there's a difference in the sensor output. And when we look at online database of potentially pathogenic variants that has been found in clinical samples, we determined that about 5% of them is already compatible with this kind of differentiation. Okay. Another type of the change of identity for transcripts is gene fusions, especially common found in cancer driving mutations. And because we already have this split design, right, we ask ourselves, can this split design be a model for detecting fusion genes? So this would be, now the bottom trigger is a fused gene, and the pre-fusion copies are represented by these two control constructs, which together still form all these double-strand regions, except that now there are two separate transcripts, right? So when we compare our sensor response to this fused trigger versus the original separate trigger, we saw that indeed the signal in response to the fused trigger is much, much higher, suggests that it might be possible to use radar sensors detecting gene fusions for future applications, such as detecting cancer cells. Okay, and now I'm going to take one last surprising turn at for the last slide, which is our neighbors happen to be great plant biologists. So we ask ourselves, if we do believe that the radar mechanism is as straightforward as we think, it's self-contained, right? It takes two RNA strands to form a double strand, and it takes the ADAR enzyme to edit them. Plants actually do not have ADAR, but we predict that if you just take our constructs and overexpress ADAR in plants, the sensor might still work. So again, there's no optimization. We just took our constructs, switched them to plant promoters and plant vectors, put them into the leaves of tobacco plants, and the data looks like this. So here again, the cherry is the co-transfection marker, and EGFP is the radar output. On the closed circles are those co-injected with a matched trigger, and the open circles are those co-injected with a mismatched trigger. So what I'm hoping you would appreciate is the color is greener, the green is higher when you have a trigger versus when you don't have a trigger, right? It's a more yellowish hue here and greenish hue here. My apologies to the colorblind people who are watching this, but we also quantify this, right? So this, the horizontal axis is the co-transfection marker, and this is signal. And as you can see, there's a trigger-dependent increase of signal in these conditions, and that made us quite happy in the sense that now we believe for pretty much any organism, right? If there's a reason to optimize this, we can potentially build you a radar sensor for it. Okay. And everything I just shared with you is already in a preprint. You're welcome to read about it. And Eric also pre-generated potential sensor sequences for human mouse transcripts. It's publicly available either in a searchable interface, or you can download the whole data set. And if you have a favorite organism that you want to detect, feel free to reach out to me or Eric, and we're happy to run the bioinformatics pipeline and generate potential sensor sequences for you and upload them to the website. And now I forgot I have this visual here. I apologize for not replacing Uncle Sam with Auntie Sam. I'm not particularly proud of Uncle Sam as of last week. But who I really am proud of are these group of young people who joined my lab. I started my lab over 2 years ago in the middle of the first COVID shutdown, right? But despite that, they have already achieved so much already, and I'm so thankful to them. And finally, I also want to thank the funding agencies, and as you can see, NIH and IBIV is over there on top of the list. Yeah, that's all I have. Thank you for the 2 people who stayed, and I'm ready to take questions if either of you have any.
Wonderful. So, David, do you have any comment or challenging question? I have question, but I want to wait your one, and then I'll ask my question.
Sure, I got just maybe one quick question, and I will admit my attention was taken away from this for a few minutes. So maybe you did cover something, but I guess when I think about synthetic biology, right? There's this sense and respond kind of paradigm that exists a lot. So you talked a lot about the sensing, right? Mm-hmm. What's first on the list of things to respond to? Like where might this go to start, closing a loop?
Yes. And by the way, I'm having trouble stopping sharing my screen. Oh yeah. How do I do that?
That should be okay. So there you go.
All right. Here we go. Yes, right. I mean, respond. Right. So the question I think is if we can detect the cell state, what's the output, right?
Yeah. Like what's your first Yeah, output
Yes.
that you would go
No,
after.
that's great. That's great. That saves us one phone call because that's basically what's in that trailblazer I told you about, right? So there are these usual things we go to as synthetic biologists, and specifically in that one, what I was saying, I know it's publicly recorded, but it's fine. I think it's not. It's quite obvious, which is if you can detect the cancer cell, right? You can say, well, now you can output anything you want. In my old work, it's a caste-based. You just kill these cancer cells. But then people correctly point out that killing cancer cells is not smart because with synthetic biology, you cannot deliver into 100% of cancer cells, right? You kill the ones you deliver into, and the ones that you don't deliver into come back. So the next thing we want to release are immune system engaging things, right? Including cell surface markers to tell the T cell to eat them, and also cytokines to tell the T cell to tell other cells to come over and eat more of the neighboring cells, right? So that's the next thing we want to try. But that's the very first I envision of a long list of things we can do, and we can get fancier and fancier, right? So for example, one thing, again, a favorite reframe of synthetic biologists is you direct the differentiation of cells down the Waddington landscape, right? So the idea is right now you want to go from cell type A to cell type B, you jam the steering wheel, and you have like 1 billion cars, and some of the cars will accidentally hit the destination. But the right way to do it is to steer as you go. And previously, people steer as you go using like transcriptional responses, but actually that's very hard to engineer in a mammalian cell, right? You have to have a transcription factor, and you have an enhancer, and you put them into a mammalian cell and still hope that in every cell they work the same way despite epigenetic and stuff. But now what we can envision is to have a piece of RNA vector, right? And that encodes the entire radar sequence of events. So the first one will be, sensor A in response to reprogramming, for example, and then turns on transcription factor B and C, and sensor D in response to intermediate cell type, then turn on another transcription factor. And the great thing about this is all of this is now just on one piece of RNA, right? So it's transient. There's really no scar after the job is done. The whole thing can be degraded. And also it's much more, we have not proven that yet, but we hope it's much more programmable because all you need is to take a 90 base pair sequence in response to a favorite transcript, and that's it. Right? So one thing I did not emphasize because I did not want to rub Taesuk the wrong way. The one thing I really, enhanced engineering, maybe very sophisticated E. coli, but it's still very hard in mammalian cells, right? For your favorite marker gene, traditionally people try to mimic the expression pattern using an enhancer, and it's still half science, half art. But right now we don't have to engineer anything. You literally just have to find a unique sequence in the transcript you detect. You make the reverse complementary of that, put this on a sensor, and that's it. There is no more fancy engineering, which is why we are so excited. And 3 other labs, at least, are very excited about it as well.
Yeah, thanks.
Fantastic. I mean, this is amazing talk. So you know what? I'm still stay in bacteria because I'm not brave enough to enter mammalian cell areas. I mean, you are brave enough to enter that one early enough. So I'm fascinated by your talk. And my question is, I'm so excited because today it's very interesting. This is coincidence or fate. I just listened to Jay Kisling's brilliant talk about anti-cancer drug making, total 30-step pathway. That was amazing. And then you're also talking about a little bit about medical application and so on. And then now currently the Jim Swartz, the same university, your department professor, very famous one, and so giving a talk right now. So and this probably I also need to call today is the Stanford University Day or whatever. So anyway, so my question is, so what would be the remaining challenges and opportunity to consider this field or make this field do a quantum jump? Because I mean, you mentioned this is still very challenging, because I mean, I understand because that I start to do some project in mammalian system because I believe that is very important for medicine. But still, that's to me, I mean, very, very challenging because there are many unknown, still many challenges, but still very huge opportunity we need to pursue. But somebody kind of do some quantum jump kind of development of thing, the biology engineering biology much easier. So I mean, I hope you do something, but I just want to hear your insight. What could be, yeah.
No, thank you. That's probably way above my pay grade, but I can share what I have, right? So I know you asked about field, but I can start with our sensor and use that as example, right?
Sure.
So one obvious problem right now is we all claim we do 0 to 1 engineering, but in reality, I would claim all these papers, including ours, are more like 0 to 0.1 engineering, in the sense that it's not quite ready for prime time yet. It can detect certain things bespoke if you happen to have a good sensor, but we don't fully understand. And I think the challenge here, I don't know what's the right motivation, right? Because the first paper you got 0 to 0.1, you get a flashy paper and grants and stuff, right? And then all the subsequent ones usually get dwindling return on this, and the motivation for making this better is diminished. Unless you have, in this case, I think we'll just keep hammering at this because we believe it's broader impact, but you never know, right? Because I can imagine many of these tools actually would have been awesome if people keep working on them, but they have one cool demonstration and then it's moved on. I think that's a general problem, but maybe that's a general problem in academia, I don't know. But the bigger challenge that I'm not directly involved in, but I would very much like to be solved by some other quantum leaps, AA is delivery, right? And everyone talks about this, and especially about synthetic biology, without good delivery. Right now, we're trying to be smart. We pick candidates where either there are some feasible, in vivo delivery that's at least in the pipeline, preclinical pipeline, or maybe it's some extracellular cell engineering, such as adoptive cell therapy, we can latch onto, right? So the idea, before circuits are medicines, we want to use circuits to improve existing cellular-based medicine, right? But hopefully that, because in principle, any cell therapy can be converted into a gene therapy if your gene delivery is efficient and specific enough. Does that make sense? You don't have to take the T cell out, engineer and put that back in, right? You just have a genetic vector goes exactly to the right T cells and do the thing. That would be my dream, but it takes many different fields to get there. And the other big problem we constantly have, especially in the immunogenicity project I'm not talking about, is the fundamental catch of doing human research without being able to test this in humans, right? Because if it's not good enough, you cannot test it in humans. But if you cannot test in humans, especially for immunogenicity, you never know whether it's good enough. And that chicken and egg, people try to deal with this with organoids, and there are a lot of progress, but then I don't know how far organoids can take us, right? Because after all, it's organoid. If organoid is too much like a human, then there become ethical issues. So I don't know if organoids can recapitulate everything you want to learn about humans. And then everybody will tell you mouse is not a human, right? So there, for delivery, I can at least imagine some future progress for a human predictive testing, a test bed, right? For mammalian synthetic biology. I very much hope somebody will solve that. But
Yeah, thank you so much. I believe my question is broad and high level, but your answer is very, very specific or to the point. And so that you said you are not in pay grade, but you are the leaders, future leaders. So you should give the vision to us. So thank you so much for your wonderful answer. So it's already 11:08 my time, so I need to close. So thank all for joining and staying today, especially when I heard from many of my friends, they actually on the road for the holiday. We have the exciting holiday soon in US. So we will meet again next week on July 7 after the holiday, Independence holiday, the Thursday, the same time, the same Zoom link. But I will be joining from London, UK after my 20th talk at Imperial College London. We'll have Dr. Greg Beckham at National Renewable Energy Laboratory and Professor Dave Turung at NYU. And basically, I'm thank you for everybody, especially the two speakers today. I'm staying until this time. And then I also thank everybody who joined, and then also thank everybody who will watch this video. And I will stop recording. In that way, we can speak more freely. Let me stop. Okay.